Dear students, good morning to all. Hope you all are fine. Today's our offline class is about some of the transgenic animals. You already know what transgenic animals are. Transgenic animals are those animals which carry foreign genes. Examples of transgenic animals. Some important examples of transgenic animals are as under. Number first, transgenic fish. Gene transfers have been successful in various fish such as common carp, rainbow trout, Atlantic salmon, cuttlefish, catfish, goldfish, zebrafish, etc. Now, transgenic salmon. Genetically modified salmon was the first transgenic animal for food production. The genetically modified sperms were fused with ova of the same species. The zygotes, which developed into embryos, gave rise to much bigger adults than either parent. The transgenic salmon possesses an additional gene that codes for the growth hormone that allows the fish to grow larger more rapidly than the non transgenic salmon. Now, another example of transgenic animal is transgenic chicken. Avian leukosis virus or ALV is a serious viral pathogen of chickens. D.W. Salter and L.B. Crittenden in the year 1988 have produced an ALV resistant strain of the chicken by introducing a defective genome of this virus into the genome of the chicken. This principle is also applied to evolve transgenic fish that can resist viral infections. Now, transgenic mice. Mouse is the most preferred mammal for studies on gene transfers due to its many favorable features like short oestrus cycle and gestation period, relatively short generation time, production of several offspring per pregnancy, that is litter, convenient in vitro fertilization, successful culture of embryo in vitro, etc. As a result, the techniques for gene transfer and transgenic production have been developed using mice as models in other animals. Recently, rats and rabbits are being used for research work on gene transfer. Now, another example of transgenic animal, that is transgenic rabbits. Rabbits are quite promising for gene farming or molecular farming, which aims at the production of recoverable quantities of pharmaceutically or biologically important proteins encoded by the transgenes. The following human genes encoding valuable proteins have been transferred into rabbits: interleukin 2, growth hormone, tissue plasminogen activator, alpha 1 antitrypsin, etc. These genes were expressed in the mammary tissues and their proteins were harvested from milk. Now, transgenic goats. Goats are being evaluated as bioreactors. Some human genes have been introduced into goats and their expression achieved in memory tissues. This initial the initial results are encouraging. Now transgenic sheep. Transgenic sheep have been produced to achieve better growth and meat production. For example, human genes for blood clotting factor IX and for alpha 1 antitrypsin have been transferred in sheep and expressed in memory tissue. This was achieved by fusing the genes with the memory tissue specific promoter of the bovine beta lactoglobulin gene. Human growth hormone gene has also been introduced in sheep in order to promote growth and meat production. However, they also showed several undesirable effects like joint pathology, skeletal defects, gastric ulcers, infertility, etc. In 1990, Tracy, the transgenic ewe, was born in Scotland. Now, the transgenic pigs. The rate of transgenic production in pigs, sheep, cattle, and goat is much lower, usually less than 1%, than that in mice, usually between 3 to 6%. The objectives in transgenic swine, plural, same meaning pig, production are number one, increased growth and meat production, and number second, to serve as bioreactors. Transgenic pigs expressing human growth hormone do show improved growth and meat production, but they also show several health problems. In January 2002, an Edinburgh based therapeutics committee announced the birth of a litter of transgenic pig clones, now transgenic cows. The only successful transfection technique in cows is microinjection of fertilizer ova, which may either be recovered surgically or may be obtained from ovaries extracted from slaughtered cows and cultured in vitro. The two chief objectives of transgenic production are as follows. Number one, increased milk or meat production, and number two, molecular farming. Several human genes have been successfully transferred in cows and expressed the memory tissue. The protein is secreted in milk from where it is easily harvested. The name of first transgenic cow is Rosie. Now, transgenic dogs. Doggy is a transgenic dog with excellent smelling power. It was used during attack on World Trade Center WTC of the USA in 2001 to recover injured people from heaps of devastating building. Andy. DNA of a fluorescent jellyfish was introduced into an unfertilized egg of a rhesus monkey in the test tube. The diploid egg underwent cleavage and the early embryo was implanted in a surrogate mother. Andy, The first transgenic monkey was born on October 2, 2000. It has been named Andy, the acronym of inserted DNA. The credit for production of Andy goes to Dr. Gerald Saccheton of Oregon Health Sciences University, USA. This work would be helpful for curing diseases such as breast cancer, Alzheimer's disease, diabetes, and AIDS. This was about some of the examples of transgenic animals.